Hello everyone, this is the first Senior Vital cast on cancer nanotheranostics. My name is Yaser Gholami. I'm a postdoctoral researcher at the School of Physics at the University of Sydney, and I've been a Senior Vital member for 4 years now. I actually became a member when I was a PhD student, and I received the Senior Vital Research Scholar Awards then, which enabled me to travel to Harvard University and gave me the opportunity to present our research to the world expert in the field of nanoscience and nanotechnology, and also further to promote our international collaboration with top research groups. Well, today's episode focuses on my research project, which is on developing a novel theranostic nano-platform for simultaneous multimodal imaging and radionuclide therapy of lymph node metastases. The accurate characterization and treatment of lymph node metastases is one of the most important topics in the field of medicine. Why? Well, metastasis is the leading cause of death from all cancer types and first appears in tumor-draining lymph nodes. For many patients, by the time the primary cancer is detected, it has already metastasized. This is further compounded by very limited and largely ineffective treatment strategies, resulting in poor prognosis across all cancer types. And indeed, 90% of patients succumb to metastasis within 5 years of diagnosis. Because of their small size and low vascularization, metastases are very difficult to detect, yet the extent of metastases in tumor-draining lymph nodes is the most important prognostic indicator of clinical outcome. Even when metastasis is detected, the high dispersion of metastatic tumor cells challenges conventional treatment strategies. To date, an effective and safe method capable of exclusively targeting metastatic tumor cells that have spread to the lymph nodes or other organs does not exist. In addition, existing imaging modalities such as PET, SPECT, and MRI have their own separate advantages and limitations in detecting lymph node metastases. For example, MRI provides good spatial resolutions, but it has a poor sensitivity, and vice versa for PET and SPECT imaging modalities. So, considering this unmet clinical need, a number of nanoparticles have been approved for clinical use to enhance imaging, in particular magnetic resonance imaging, where contrast can be enhanced by magnetic nanoparticles that induce a strong nanoscale magnetic field inhomogeneities within tissues. Further studies also have shown a radiolabeled nanoplatform that can combine PET and MR imaging modalities in a complementary manner could potentially even result in a higher accuracy of detecting micrometastases or small metastases within lymph nodes. In cancer nanomedicine, various multimodal imaging and drug delivery techniques have been developed based on nanoparticles, which, owing to their nanoscale size, can interact effectively at the molecular level with tumor cells. To date, however, the majority of imaging or therapeutic nanoparticles platforms rely on chemical chelators to target, image, and deliver drugs to tumor cells. This is problematic because different chelators are needed for each of these capabilities, and when tested in vivo, many are found to be unstable or even ineffective despite promising in vitro studies. Furthermore, most of cancer nanomedicines developed to date have neglected to address the primary cause of cancer mortality, metastasis. So, to address all these issues, we have developed a chelate-free theranostic magnetic nano-platform for non-invasively detecting, diagnosing, and eliminating lymph node metastases. We have used the FDA-approved nanoparticle called Ferraheme as a nano-platform. We have successfully radiolabeled Ferraheme nanoparticles with two common PET tracers, copper-64 and zirconium-89. This enables simultaneous multimodal imaging of copper-64 and zirconium-89 Ferraheme with hybrid MR PET scanner. And for example, when this radionanomedicine is injected to a patient, monocyte tracking delivers radiolabeled nanoparticles to the lymph nodes where metastatic tumor cells can be detected with PET and imaged with MRI. And our radionanomedicine platform is going to be the first to exploit the simultaneous MR-PET imaging technologies for detecting and diagnosing lymph node metastases. Additionally, we have developed a chelate-free therapeutic nanomedicines by radiolabeling ferriheme nanoparticles with the therapeutic isotopes such as yttrium-90 and lutetium-177 with high radiochemical yield and purity. Following a positive diagnosis of lymph node metastases, yttrium-90 ferriheme and lutetium-177 ferriheme can deliver targeted internal radiotherapy to tumor cells. Interestingly, as yttrium-90 and lutetium-177 isotopes can be also imaged with PET and SPECT respectively, yttrium-90 ferriheme and lutetium-177 ferriheme are theranostic radionanomedicine that enable PET or SPECT imaging in conjunction with MRI for image-guided targeted delivery of radionuclide therapy and also to monitor treatment response. So, potentially, this multifunctional radionanomedicine platform can be used to detect, diagnose, and treat lymph node metastases arising from any primary cancer site, such as colorectal, breast, prostate, lung, and liver. We are currently performing animal studies using our developed radionanomedicines, and hopefully these studies will led us to human clinical translation in the near future. In conclusion, I think the ability to non-invasively detect, diagnose, and eliminate lymph node metastases using immunoresponsive radiolabeled nanoparticles could represent a paradigm shift for cancer medicine in the 21st century. And we hope that with the development in nanomedicine, we can have a small contribution towards helping patients with cancer and doing something that our society as well as humanity can benefit from it. Thank you.